So hello students and welcome back to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about gene, that what is a gene and how does the gene act in the cell. See, previously there were several different definitions of the gene. In our class 12th and in our graduation, we were being told a gene is a segment of DNA that leads to the formation of protein. So gene was considered to be a DNA fragment that would lead to the formation of a particular protein. Okay, so this will lead to the formation of an mRNA, and mRNA will lead to the formation of a protein. But it is not true. A gene can be any segment of DNA which performs any function. A segment of DNA which performs any function could be a gene. Why? Now, see, what about transposons? Transposons are also referred to as jumping gene. Transposons are also referred to as jumping genes. These genes have the ability to jump from one location to the other, and they can move from one location to the other, but they will never be translated into a functional protein. And it is not 100% sure that every time they would be transcribing and would be forming a protein. So they are referred to as selfish DNA. These are referred to as selfish DNA molecules. Okay, so these are the molecules which are present, and these are referred to as selfish DNA molecules. So they are referred to as transposons, but transposons are also referred to as jumping genes. That means these are being considered to be a part of the genes. Okay, so these are jumping genes in nature. Okay, so these are jumping genes and are referred to as transposons. The same is true for other different types of genetically sequences. For genetic sequences, for example, when you talk about promoters, as you know, these are cis-acting DNA sequences which are necessarily required for the accurate and effective transcription of the gene. These are cis-acting sequences sequences which are required for the accurate and effective transcription of a particular gene, but they will never be translated into protein. So they are not going to lead to the formation of protein, but they must be required, and they are essentially required in order to form a functional gene. So they would be constituting a very important part of the gene functional gene is being made by these only. So a gene has been made functional only in the presence of these promoters. So all of these are being said to be a part of the gene. These are all the part of the genes which are present. Okay, so how can you define a gene? A gene can be defined as a DNA sequence that encode protein variant produced by post-translational processes other than RNA splicing or RNA editing. Now, recently, the modern definition of the gene being that a gene now is referred to as a segment of DNA that lead to the formation of that performs a specific function. A gene refers to a segment of DNA, refers to a segment of DNA, refers to a segment of DNA, a gene refers to a segment of DNA, segment of DNA. A gene refers to a segment of DNA that performs a specific function, performs a specific function, specific function. So this is a segment of DNA that performs a specific function is referred to as a gene. So this would be referred to as a gene. Now, in the case of splicing, so this is the accurate and the most modern definition of the gene. So any segment of the DNA that performs any function will be referred to as a gene. Okay, this is the new and the modern definition of a gene. Now, what do you mean by splicing? What is splicing? Trans-splicing is another variation of splicing process. In this type of splicing, the two different types of genomic sequences, the two different types of introns are being spliced in such a manner that the exon 1 and exon 2 are ligated together. This is exon 1, this is exon 2. The exon 1 and exon 2 are going to be ligated together in such a manner that these are the two sequences that have been joined. This is exon 1, this is exon 2. They have been joined with regard to each other. Okay, and the intronic sequences which are present has been removed. This is the intron that has been removed. Now, this is a Y-shaped. branched intronic structure. This is the intron which has been removed. Intron removed. When you look at this structure is a Y-shaped structure as compared to the lariat type structure that was present in the presence of adenine. There was a structure which was present, and this structure was the lariat type structure which was spliced off in the case of group second introns, and in the case of your spliceosome mediated intron splicing, spliceosome mediated intron splicing. Intron splicing was involving the formation of a lariat like structure. Lariat structure. This one was lariat structure which was being produced. This one was the lariat structure which was being produced, and this lariat structure was being removed. Now, instead of this formation of this lariat like structure and the formation of this lysosomal complex as well as the group second intron, here what we get is that these two exons and introns, they are being formed from different types of an hnRNA. They are being formed from hnRNA, different types of hnRNA encoded by different genes. For example, this one was first one was being encoded by gene 1. Okay, gene 1 located on a separate homologous chromosome. Another is being encoded by the gene second, located on the second homologous chromosome, which is located on another chromosome, which is not homologous. Now, these two genes which are present here, these two genes have different sequences present within itself. They have introns and they have exons present. Now, this is referred to as the intronic sequence, the non-coding sequence. So this is non-coding. This is non-coding. This is non-coding, and this sequence which is present is referred to as coding. Okay, so how does splicing takes place? Splicing takes place in a manner that the exon 1 guanine residue, exon 1 guanine residue and the uracil residue, here the guanine is going to be bounded with the branch point adenine. This adenine will have a hydroxyl group present over itself. This adenine will have an OH group present over itself. When this adenine will have a hydroxyl group present over itself, this hydroxyl group would be attacking the guanine molecule. It would be attacking this particular, sorry, just a second. It would be attacking this guanine molecule, okay, in this manner. This attack would be mediated by the hydroxyl group. When it will attack the guanine molecule, it will cause the ligation of the guanine molecule, and the formation of this Y-shaped structure is going to take place. So this is the Y-shaped structure produced. Here adenine is present and it has been bounded with this guanine molecule. And here the ligation has taken place. Okay. Here the ligation has taken place, and this is it. Adenine has been linked with the guanine residues. There it has been bounded. Okay. Now, further reactions takes place in such a manner that the reaction which have occurred here, this reaction will cause the release of the 3 prime from the exon 1. This is exon 2. This is exon 2. Okay. The exon 1 has been released. When the exon 1 has been released, then what is going to happen? The exon 1 is going to work here. In what manner? This is the exon 1. This is the exon 1 that would be working here. And this exon 1, just a second, will be cleaved here. In what manner it would be cleaved? That we will have to see, and how it would be cleaved. Cleaving the reactions. Now, exon 1. Exon 1 has its own 5 prime end. It has its own 3 prime end. Now, this 3 prime end will make an attack over the guanine residue. It would be making an attack. How? In this manner. When it will make an attack, the exon 1 and exon 2 are going to be ligated together. When exon 1 and exon 2 are going to be ligated together, these two are going to ligate and they will lead to the formation of a protein. These could be translated into a protein, translated to form protein, translated, related to form proteins, having the exon 1 part as well as having the exon 2 part. This is how a complex protein is being produced. This is exon 1 and this is exon 2. Exon 1 and exon 2 both are going to work and both are going to be ligated. Any doubt, any problem? So this type of splicing takes place in a very specialized RNA, which is referred to as slRNA. Trans splicing occurs in the case of slRNA. Trans splicing, which has been discovered in few eukaryotic organisms like single cell trypanosomas. So it has taken place in trypanosomas gambiense, the grand old man of intestine. Exon from two separate RNA transcripts are spliced together. See, exons from the two separate RNA are being spliced together to form a mature mRNA molecule. It will lead to the formation of a mature mRNA molecule in trypanosoma. A single exon is spliced onto the 5' end of many different RNA transcripts produced by the cell. In this way, all the products of trans splicing have the same 5' exon and different 3' exons. So the 5' exon, this particular exon will be same in all. So this would be same in all, but the 3' exon is going to be different. So 5' exon, this exon is going to be same for all, but 3' is going to be different. 5' exon will be same for all, will be same for all, will be same for all, same for all, while 3' will be different, while 3' will be different, will be different, will be different. This is how splicing takes place. In this manner, the splicing is going to take place. And such type of splicing is referred to as trans splicing. See, it occurs in a specialized RNA called as slRNA. Spliced leader RNA molecule provides an exon that is connected to the first exon of a mature mRNA, and the splicing is referred to as trans splicing. Okay. So this is the Y-shaped lariat that is being produced. Then trans-splicing, two exons initially found in two separate RNA molecule, two exons which have been produced from two separate RNA molecule are spliced together into a single messenger RNA. There is a single messenger RNA in which it has been spliced off. A Y-shaped branch structure has been produced. This is because the initial reaction bring together to two RNA molecules closer to each other, forming a loop within a single molecule. Next is referred to as self-splicing introns. Now, self-splicing introns are the one that have the ability to splice themselves from the RNA. Okay, these are also referred to as ribozymes. Ribozymes are the molecules when RNA act as an enzyme. When RNA act as an enzyme, it is referred to as a ribozyme. When RNA act as an enzyme, act as an enzyme, it is referred to as a ribozyme. When RNA acts as an enzyme, then it is referred to as a ribozyme in nature. Okay. So group 1 introns and group 2 introns are two distinct classes of self-splicing RNA molecule that contain their own active site. So group 1 introns as well as group 2 introns, they have their own active site for intron removal as well as for exon ligation. Here the introns are being removed and the exons are going to be ligated together. The introns will be spliced and the exons will be joined together with each other. Now, how the reaction occurs, that we have to see. These two classes of ribozymes are distinguished by their mechanism of splicing. In the first group 1 splicing, you find these types of the introns have a branch point guanine. The branch point guanine has an hydroxyl group present over itself. This hydroxyl group will attack the first exon. This is the exon. This is the exon number 1. This is exon number 1, and will be attacking the exon so that the exon will get separated. The exon, the free exon 3' OH, the free exon 3' OH, that is free. This is 5', and then this is 3'. This is the 3' OH that has been made free will be attacking the guanosine residue. Once it would be attacking the guanosine residue, then what is going to happen? Then the reaction is going to take place, and this guanosine is going to be replaced. Here the bonding will be broken down. The exon 1 and exon 2 would be ligated together. This is exon 1, and this is exon 2. So exon 1 and exon 2 would be ligated together with each other. This is exon 1 and this is exon 2. Both the exons have been ligated together. Both the exons have been ligated together and the intron has been removed. This is the intron that has been specifically removed, and then it will undergo cleavage and degradation. This is the intron that would be cleaved and degraded. In the group second introns, the same mechanism operate, but in a slightly different manner. In the group second introns, you have a branch point adenine. This adenine is commonly referred to as the branch point adenine. Branch point adenine, adenine. Here there was a branch point guanine, branch point guanine. Okay, this branch point adenine has an hydroxyl group. That hydroxyl group is going to attack, and the exon 1 and exon 2 are going to be joined together with each other. This is exon 1 and this is exon 2. Exon 1 and exon 2 are going to be ligated together with each other. When the exon 1 and exon 2 are going to be ligated together, and the intronic region would be removed. See this exon 1, the free 3' hydroxy group of the exon 1 will attack the intron exon boundary. In the case of exon second, both the exons will be ligated together. This would be the 5' end, and this would become the 3' end. Both the exons are being ligated together. Here it would be the 5' end, and here it would be the 3' end. 5', 3', 3' and 5' are being joined together with each other. This is 5' and this is 3'. So this is exons. So here you can find this is 5', this is 3', this is 5', and this is 3'. So here you have the two exons ligated together. This is referred to as exon 1 and this is referred to as exon 2. Okay, and here you have this lariat like structure which has been removed and will be degraded. This is the part of the RNA that will be further degraded. Okay, so there are the splice sites which have been produced, and this is how the process of splicing will take place in the case of intron splicing. Now, what we will have to study is that how the process of homing and retrohoming takes place. What is homing and what is retrohoming? So here, from here, we will have to start about discussing about the process of homing and retrohoming. You can look here. The group 1 intron sequence begins the splicing reaction by binding a guanine residue to the intron sequences. There is a guanine residue that has been present in the intronic sequences. The guanine is thereby activated to form the attacking group. Guanine gets activated, forming the attacking group that will break the first of the phosphodiester bond, cleave during splicing. It will cleave the first phosphodiester bond. So this is the first phosphodiester bond that would be cleaved. In group second intron sequences, a specialized reaction residue, adenine residue in the intron sequence is the attacking group, and a lariat intermediate is being generated. Then in the next reaction, you can find that the first exon hydroxy group will attack the exon number second. When the hydroxy group will attack the exon number second, then a lariat shaped structure will be removed, and this is the lariat shaped structure that would be subjected to degradation. Might be degraded. It is not always that it is degraded. Sometimes the RNA molecule that has been removed can move to new locations and can even integrate into the genomic DNA. Then it would be referred to as homing and retrohoming. Now see this. Unlike the group 1 introns and group second introns utilize an internal nucleophilic attack for the first, the OH of a highly conserved adenine residue is going to take part in the case of intron splicing in the group second introns. In the group second introns, you find that there is an adenine residue that is involved. See, both group 1 and group second introns are metalloenzymes. Both of these intron splicing processes are being mediated by metalloenzymes. Metalloenzymes are the enzymes that have a metal ion present within their active site. Metalloenzyme means metal ion constituted enzymes. Metal ion constituted enzyme. Constituted enzymes. Okay. So these are metalloenzymes. They have a metal ion present over themselves, which require divalent metal ions for activity. Although the metal specificity varies subsequently among group 1 introns, it is usually satisfied by magnesium ion and manganese ion. So metalloenzymes functions with magnesium and manganese ions. Many group 1 introns in organelle genes contains an open reading frame which codes for a protein called as maturases. Okay, so group 1 introns contains an open, in fact sometimes the group 1 introns which are present in organelle may even contain an open reading frame. That means it could encode for the formation of protein and encodes for a protein called as maturase that appears to play a role in splicing. Okay, so there is another enzyme that is present that is referred to as maturase, and this maturase helps in the process of splicing to take place. Okay, now what do you mean by maturase and how does splicing take place? Splicing will occur in the case of group 1 introns by itself because they contain an open reading frame and could lead to the formation of a protein biosynthesis. Sometimes these group 1 intron in the presence of maturase will form a particular 3D protein. This would be a 3D protein molecules that could be produced. Okay, this is a protein. This is a protein molecule that has been produced from this maturase enzyme. Okay, it has led to the formation of a protein. Secondly, in other cases, group second introns are mobile element that transpose as a remarkable process called as retrohoming. In this retrohoming, this RNA molecule that has been removed in the case of group second introns, this RNA molecule, this is the RNA, this is the RNA molecule, okay, intron, this RNA will be removed and would be reverse transcribed to form a DNA molecule. And this DNA molecule that has been produced can be inserted to a new site, inserted to a new site within the genome, inserted to new site, to new site. Just a second. Inserted to a new site. Inserted to a new site. Site on the DNA. On the DNA. So it would be inserted to a new site on the DNA. Okay. This is how the splicing will take place and retrohoming will occur. In this process of retrohoming, annexized introns comprising single-stranded RNA insert directly into an organelle genome prior to being copied into a double-stranded DNA. It would be inserted, it would be directed into an organelle genome prior to being inserted in the form of a double-stranded DNA copy. It can be converted into double-stranded DNA copy and can be inserted into the organelle genome. Now, let us have a look at the animation in order to understand the phenomena of retrohoming, in order to understand that how the group 1 splicing actually takes place. So please let us have a look at the animated version of the lecture, the group 1 introns and how
While
does
most
group 1
introns are removed,
introns work. Please have a visualization of the same.
While most introns are removed using a large complex called the spliceosome, some introns are capable of catalyzing the chemistry of their own release. These self-splicing introns are divided into two classes based on their mechanism of self-splicing: Group 1 introns and Group 2 introns. When you have completed this exercise, you should understand the structure of group 1 introns, understand the mechanism of self-splicing by group 1 introns, and understand how group 1 introns use cyclization to prevent themselves from participating in the reverse reaction.
Now, we must understand that what is group 1 intron, what is group 2nd intron, and how does group 1 introns and group 2nd introns differs from each other. We should know the mechanism of cyclization of the group 1 intron, in what manner the cyclization of the group 1 introns takes place, how the removal of specific intronic sequences occurs in the case of the group 2nd introns, where the intron is being removed in the formation of a lariat like structure. We have to understand the mechanism of splicing. We will have to understand about the process of cyclization that takes place in the case of group 1 introns.
The first group 1 intron was identified in the ribosomal RNA of Tetrahymena thermophila, a unicellular ciliated freshwater protozoan. In fact, this group 1 intron was the first RNA molecule proven to be capable of catalyzing a chemical reaction without the aid of proteins. Such catalytic RNA molecules are called ribozymes.
So, the first group 1 introns which were being known, which were being identified in the case of Tetrahymena thermophilus. Tetrahymena thermophilus is a ciliated protozoan which has the ability to survive at high thermophilic. It is thermophilic, so it can survive at high temperature environmental condition. Okay, it is a protozoan. It is a protozoan which is ciliated. Next, now this here, the first ribozyme was being discovered. You can look here. It was being discovered by Thomas R. Chase, who had discovered the first ribozyme. Please let us look forward.
Group 1 introns are capable of folding back onto themselves, forming a complex secondary structure.
So they can fold back over itself and leading to the formation of a complex secondary structure within themselves. Next. So this is the group 1 introns in structure that you can visualize. So they would be forming a circuit type structure.
This secondary structure can further fold into a tertiary structure. This tertiary structure contains two important features: a guanine binding pocket and an internal guide sequence. The internal guide sequence is capable of base pairing with the exon at the 5' end of the RNA molecule.
So what you can find here is that there is a guanine binding pocket that is present. This guanine binding pocket is the site where the guanine nucleotides can specifically bind. Now see, group 1 introns differs from group 2nd introns because group 2nd introns were having a branch point adenine present within their structure or a branch point guanine present within their structure. Okay, here there is no GUAG or AUAG consensus sequences or conserved sequences present. Here there is a internal gap region which is present, and this region has the ability to bind to guanine from the cytosol, and that is why this pocket is commonly called as guanine binding pocket. Okay, so this pocket will be called as the guanine binding pocket. It will initiate the reactions to take place. Internal guide sequences binds with the exon 1.
The guanine binding pocket is capable of binding any guanine containing ribonucleotide, including guanosine, GMP, GDP, GTP, or an RNA molecule that terminates in a guanine residue.
So it can bind with any molecule. It can bind with GDP, GTP, or guanosine, or any molecule that can terminate with the guanine residue. Next.
To begin self-splicing, a free guanine containing ribonucleotide binds in the guanine binding pocket. The 3' hydroxyl of this guanosine attacks the phosphate at the 5' end of the intron.
So see this. In this reaction, what is going to happen? Firstly, to the guanine binding pocket, a free guanine molecule has been bounded. Now, this free guanine molecule that has been bounded will be attacking the splicing site, the splice site, the 3 prime splice site of the intron. Here you can see that it is this site, the 3 prime is end of the exon towards the 3 prime end of the exon 1, or towards the 5 prime splice site of the intron. The binding takes place and the introns are being removed.
This transesterification reaction fuses the guanosine to the 5' free end of the intron.
So this is the transesterification reaction that is going to take place, and the transesterification reaction is going to fuse this guanine to the free 5 prime end of the intron, and now the reaction is going to begin.
The freed 3' hydroxyl of the exon then attacks a phosphate at the 5' end of the other exon. This fuses the two exons and releases the intron.
So this has fused the two exons together with each other, and the intron has been released.
The internal guide sequence is also capable of base pairing with a sequence near the 5' end of the excised intron.
Now, one very important information that you must note is that in the case of the group 1 introns, see this guanine which is present, which is acting like a branch point guanine, is actually binding to the guanine binding pocket. It is actually binding to a pocket, and that pocket is called as the guanine binding pocket. Guanine binding pocket. Binding pocket. This guanine binding pocket, which is present here, where the guanine molecule has been bounded, here you can effectively find that it was at this pocket that the guanine residues have been bounded. And here, this is the pocket, this is the region where the guanine has been bounded. Okay, and then the guanine has catalyzed the reaction. So this guanine was not an integral part of the intron, but it was being bounded because of the three-dimensional structure of the intron. ये इंट्रॉन के स्ट्रक्चर की वजह से बाइंड किया था. Next point. Okay, so this is how the reaction has occurred. I have already told you in my previous slide that in what manner the reaction has occurred. See, the guanine molecule was coming at the guanine binding pocket. The cleavage was taking place. The first axon was being cleaved by the joining of the intron and the guanine residue together with each other. The cleavage of the phosphodiester bond was taking place. Now the exon number 1, which is free, its 3' hydroxy group is free. Its 3' hydroxy group will be attacking the exon number 2. Once the exon number 2 is being attacked, the exon number 1 and exon number 2 are going to be ligated and were going to be removed off. The intron is being removed from this particular structure. In the next point, internal guide sequences base pairs with the 5' end of the intron. Now, how does base pairing and its circularization takes place? So let us have a view.
The intron also has a guanosine at the 3' terminus. This guanosine can bind in the guanine binding pocket.
Okay, so there is a guanosine present at the 3' end. This guanosine will be binding to the guanine binding pocket, facilitating the next step of the reaction.
The 3' hydroxyl of the bound guanosine attacks a phosphate to the right of the internal guide sequence, using the same transesterification mechanism used to splice the exons. This reaction is called cyclization.
Now, this reaction is referred to as cyclization because here the cyclic structure has been produced. The internal guide sequences forms base pair with the 5' end of the intron. The 3' guanosine binds in guanine binding pocket. The hydroxy group of bound guanosine attacks the phosphate at the right of the internal guide sequences. Once it has attacked to in the internal guide sequences, then the cyclization of this intron would have taken place. What prevents the guanosine at the 5' end of the intron from binding in the guanine binding pocket and attaching a phosphate group? Because of the structure, the 5' guanine was far away as compared to the 3'
The 3'
guanine. So you can look here, the 3' guanine is much more closely attached, and that is why it can bind with the guanine binding pocket as compared to the 5' guanine.
hydroxyl of the bound
Just a second, moving back
the
to
new
the
bond
next.
between
The new bonds which are being produced here.
Between the 3' guanosine and a phosphate at the 5' end of the intron is easily hydrolyzed. However, since the intron has been cleaved, the intron is unable to fold properly and catalyze the reverse reaction to splice itself back between the exons.
Okay, because it has been cleaved, so it cannot be spliced away and cannot reform the structure. Next point.
Group 1 introns have been widely studied for potential therapeutic and experimental uses.
So they
In
have
this section, you will explore some of these possible uses.
so these have been extensively studied in large number of uses in large number of therapeutics. So how they are being used, that we have to understand. The group 1 introns. First question.
Question 1. Since group 1 introns have a turnover number of 1, they are not technically enzymes. Which of the following would be necessary to convert a group 1 intron into a true enzyme?
Since group 1 introns have a turnover number that is equal to 1, because see, whenever the group 1 introns are going to work, then the group 1 introns converts only a single uh, this group 1 intron into a particular product. So which of the following would be necessary to convert a group 1 intron into a true enzyme? So where it would be converted into a true enzyme, we will have to cleave it, select on the correct location below. You may have to select more than one location. Okay. Okay, we will have to select locations like this, that where to cleave them. So now this is the exon number one, and it has to be cleaved here. Cleavage has to be done here. Another cleavage has to be done here, so that the exons could be ligated together.
Incorrect. You may
The first cleavage. See this, where from the first cleavage is going to take place. The guanine is going to cleave the first exon, and the exon 1 is going to be released, and then the exon 1 is going to cleave here. That means it should be like this. Okay, let me just check it. First cleavage and then second cleavage. This is what I did.
incorrect. You may want
See the internal guide sequence, cleave here and cleave here. Okay, sorry, just a second. The first cleavage must take place here, and then the second cleavage will take place here, in order to that for the two exons to be ligated together.
correct.
See this 5 prime, this is the 3 prime end.
Question 2. You wish to construct a ribozyme from a group 1 intron which will cleave RNA at a specific sequence. What part of the intron could you modify to change its sequence specificity?
You wish to construct a ribozyme from an intron, group 1 intron, which will cleave RNA at a specific location. You have to make an enzyme, and the enzyme is a ribozyme from the group 1 introns, which will cleave the RNA molecule. So you have to make a ribozyme that will effectively cleave the RNA molecule. Which part of the intron could you modify to change its specific its sequence specificity? Click on the correct part of the introns. You may have to select more than one answer. Okay, it is saying which correct on the correct part of the introns you will have to select. Which part of the introns you will have to cleave.
Incorrect.
So that means here. And let's
Incorrect.
check it
You
out.
may
See, the internal guide sequences base pair with the 3' end of the exon. Changes this sequence will change the target of the intron derived derived ribozymes. So what has to be done? The internal guide sequences which are base pairing with the 3' end of the 5' exon. That means internal guide sequences will be present here. Now that this molecule
incorrect.
has to be cleaved, or both has to be cleaved.
Incorrect.
Then what? Okay, I will have to look at it.
Question 3. Given this target sequence, what does the internal guide sequence of the intron-derived ribozyme need to be?
This is the target sequence. We have to click on the internal guide sequence. Okay, So the internal guide sequence. Now see this. This is 5 prime to 3 prime. So here there is the 5 prime end, there is the 3 prime end. We can look here, something like this. This is the 5 prime end that is present, and this is the 3 prime end that is present. Complementary to this, there would be a 5 prime end present here and the 3 prime end. So we have to write it in this manner. So adenine will bind with uracil, guanine will bind with cytosine, guanine will bind with cytosine, cytosine will bind with guanine, guanine will bind with cytosine, and adenine will bind with uracil. So you will have a sequence UCC GCU. Okay, just writing the answer there. So UCGCCU. Okay,
Group 1
sorry,
intron. Question 1.
written that
Group 1
answer.
intron. Group 1 intron. The intron, a free guanine. The tertiary group 1 introns are catalytic RNA.
This is
Question 6. Question 5. Question 4. Which of the following is a possible? Question 3. Question 2. You wish to construct a ribozyme from a group 1.
this we have.
Question 3. Given this target sequence, what does the internal guide sequence of the intron-derived ribozyme need to be?
It has to be something like UCGCCU. Okay, let me check this.
Correct.
Okay, so this is the sequence that has to be done. So here the splicing will take place. Here it will bind and the splicing is going to take place. This is how splicing will occur.
Question 4. Which of the following is a possible danger in this approach?
The ribozyme will likely undergo cyclization, rendering it inactive. The target sequence is relatively short, so it can appear in many different RNA sequences, not only in the desired target. The ribozyme could easily splice itself into an RNA molecule. The ribozyme would compete with native group 1 introns. Now see this. The target sequence there is very short, and such type of target sequence can appear in any of the RNA molecule, which will produce undesirable fragmentation.
Correct. Question 5. A cell contains an RNA molecule with a defective 3' sequence. How could you use a group 1 intron-derived ribozyme to repair this RNA molecule?
Now, there is a RNA molecule that has a defective sequence, and there is a defective 3' sequence. We have to use a ribozyme in order to repair this defective sequence. Use the ribozyme from question 3 to cleave the undetected defective RNA molecule. Use the ribozyme from question 3, but change the internal guide sequences. Include the correct sequence on the 3' end of the intron derived ribozyme. Include the correct sequences on the 5' end of an intron derived ribozyme. Okay, we will have to include the correct sequences on the 5' end of an intron derived ribosome on the 3' end, because this has to be removed.
Correct.
Okay, so the 3' end has to be removed. See this?
Question 6.
End
Which of
has
the
to be, this
following
is
is
the
a
correct
drawback of the
sequence, it has
method
to be
used in
there.
question 5?
Just a second. This would be the correct sequences that would be there, and the defective sequence will be replaced with the correct sequence ligation. Now,
5. To repair a defective RNA molecule.
which of the following is a drawback of the method used in question 5 to repair a defective RNA molecule? The intron derived ribozyme free require free guanosine for catalase. The intron derived ribozyme require free guanosine for catalysis. The intron derived ribozyme will also cleave DNA. No, it will not cleave DNA. The repaired RNA might include insertion or deletions. Okay, so see this. Because the repaired DNA RNA might contain insertion or deletion of certain sequences, so this would be the most appropriate one. The intron derived ribozyme can only repair one RNA molecule. Yes, it could only repair the one RNA molecule. There is only correct sequence, single correct sequence. This would be the best answer.
Correct.
Okay. See this defective RNA molecule, it will bind here. the defective would be removed and the correct sequence will be ligated together. This is the correct sequence that has been obtained. So this is how you can obtain the correct sequences. I know these questions are tough, but try to solve them up. Look at the recorded version of the lecture once again and you will get the correct
Group 1
versions of the answers.
introns are catalytic RNA molecules capable of splicing themselves out of an RNA transcript. Group 1 introns fold into complex secondary and tertiary structures.
Okay, this is the revision. They have been folded into the complex secondary as well as the tertiary structure. So these have been folded. Next.
The tertiary structure contains two important features: a guanine binding pocket and an internal guide sequence.
So it contains two different types of sequences, a guanine binding pocket and an internal guide sequence.
A free guanine-containing ribonucleotide binds in the guanine binding pocket, and the 3' hydroxyl of this guanosine attacks the phosphate at the 5' end of the intron.
So this
The
is the
freed
summary.
3' hydroxyl of the exon attacks a phosphate at the 5' end of the other exon, fusing the two exons and releasing the intron.
So this is the exon have been joined and the intron being released.
The intron then undergoes cyclization.
Cyclization and
This reaction destroys the tertiary structure of the intron, preventing it from catalyzing the back reaction to reinsert itself into the RNA transcript.
This is the back reaction.
You have completed this exercise.
So that's all. Hope you have enjoyed the lecture. The lecture is all over. You get the recorded version of the lecture at your PC where you can look at it. So that's all. We have finished intron splicing. Tomorrow onwards, we would be starting the RNA editing. Thank you. Thanks a lot. Hope you have enjoyed the lecture. That's all.